Welcome to this lecture on developmental biology. Today, we are going to explore a truly fascinating topic, the fate of germ layers. Have you ever wondered how a tiny, microscopic sphere of identical cells knows exactly how to build a complex human body with a brain, a beating heart, and a digestive system? It all comes down to a highly orchestrated biological roadmap. In this session, we will uncover how early embryonic cells are assigned their permanent career paths, and we will even look at the mathematics behind how these tissues grow so rapidly. Let us dive in. To start, let us imagine you are watching a construction crew build a massive, complex house. You know that they do not just throw bricks and wood into a random pile and hope a house appears. Instead, they lay a concrete foundation, frame the wooden walls, and finally put on a shingled roof. Each part of the finished house comes from a very specific, dedicated starting material. In biology, a developing embryo does the exact same thing to build a living body. After the initial stages of development, the embryo organizes itself into 3 distinct layers of cells. Think of it as the ultimate sorting process, where every cell is assigned a permanent career path. Now that we understand the blueprint analogy, let us look at the formal biological process. The fate of germ layers refers to the specific developmental pathways and final tissue outcomes of the cell layers formed during a process called gastrulation. During gastrulation, the embryo transforms from a simple ball of cells into a multi-layered structure. These 3 primary layers are the ectoderm, the mesoderm, and the endoderm. Every single organ and tissue in your body originates from one of these 3 layers. Together, the predictable, hardwired mapping of these 3 layers to their final adult structures is known as the fate map of the embryo. Building on this fate map, let us examine what each specific layer becomes, starting from the outside in. The ectoderm is the outermost layer of the embryo. It differentiates into your entire nervous system, including your brain and spinal cord, as well as your epidermis, or outer skin, and associated structures like hair and nails. Moving inward, we find the mesoderm, which is the middle layer. The mesoderm gives rise to the structural and fluid components of the body. This includes your musculoskeletal system, your circulatory system and blood, your kidneys, and your reproductive organs. It is the mechanical and transport infrastructure of the body. Moving to the deepest part of the embryo, we reach the final layer. The endoderm is the innermost layer of the developing embryo. While the ectoderm forms the outer covering and the mesoderm forms the structural middle, the endoderm forms the vital internal linings of your body. It develops into the lining of the digestive tract and the respiratory system. Furthermore, the endoderm gives rise to major, highly specialized internal organs, most notably the liver and the pancreas. By understanding these 3 layers, we can trace the origin of any tissue back to its specific starting point during early embryonic development. Knowing these specific tissue assignments leads us to an important question. Why does this matter in the real world? Understanding the fate of germ layers is absolutely critical because it provides the foundational blueprint for life. Without this strict, predictable organization, our bodies would just be a chaotic, non-functional mass of random tissues. In medicine, developmental biologists use this knowledge to understand, diagnose, and treat severe congenital birth defects. For example, if a baby is born with a spinal cord defect, like spina bifida, doctors know this is specifically a failure of the ectoderm layer during early development. Beyond diagnosing birth defects, this biological roadmap opens up incredible possibilities for the future of healthcare. In the modern field of regenerative medicine and stem cell therapy, scientists must replicate these exact germ layer fates in the laboratory. By guiding unspecialized cells down the specific pathways of the ectoderm, mesoderm, or endoderm, their ultimate goal is to successfully grow replacement organs for sick patients. However, to achieve their final fate, these germ layers do not just change shape, they must multiply rapidly to generate enough mass. This brings us to the mathematics of embryonic growth. To achieve their final fate, these germ layers do not just change shape, they must multiply rapidly, which brings us to the mathematics of embryonic growth. While the fate map tells us what the cells will become, we must also understand how they generate enough tissue to form a whole organ. Embryonic cells grow via mitosis, dividing from 1 into 2, 2 into 4, and so on. We model this using the exponential growth formula. N of t represents the final number of cells. N0 is the initial number of stem cells we start with. The variable t is the total developmental time elapsed, and d is the doubling time. If the initial cell count increases, the final organ size increases proportionally. If the doubling time decreases, cells divide faster, causing the population to skyrocket. Before we move on, let us see this formula in action with a concrete example. Imagine we are observing a frog embryo. We are given a small cluster of 50 mesoderm cells, destined to become a muscle group. The developmental period is exactly 120 hours, and the cells have a doubling time of 24 hours. We are asked to find the final number of muscle cells. We choose the exponential growth formula because embryonic cells double at regular intervals. We substitute 50 for the initial count, 120 for total time, and 24 for doubling time. Dividing 120 by 24 gives us 5 complete rounds of cell division. 2 raised to the power of 5 is 32. Multiplying our initial 50 cells by 32 gives us a final count of 1,600 cells. This shows how rapidly a tiny cluster forms visible muscle. Now that we have successfully calculated normal growth, let us look at a clinical scenario where development goes wrong, starting with a healthy baseline. We are given an initial cluster of 100 endoderm cells, forming a liver. The critical developmental window is 60 hours. The normal, healthy doubling time is 10 hours. We need to find the final cell count for a normal liver. We plug our values into the exponential growth formula. The exponent becomes 60 divided by 10, which means the cells will divide exactly 6 times during this window. 2 to the power of 6 is 64, which is our growth multiplier. Multiplying the initial 100 cells by 64 results in a healthy, fully formed liver primordium containing 6,400 cells. This is our baseline for healthy development. Having established our healthy baseline, let us see what happens when a genetic mutation slows down cell division. In this mutated scenario, the endoderm cells have a sluggish doubling time of 20 hours instead of 10. The initial cell count remains 100 and the time window is still 60 hours. We substitute the new doubling time of 20 into our formula. 60 divided by 20 gives us only 3 rounds of cell division. 2 to the power of 3 is 8. Multiplying our initial 100 cells by 8 gives a final count of only 800 cells. Compared to the healthy 6,400 cells, this drastic reduction explains why developmental mutations often lead to severely underdeveloped organs.
It proves how critical the timing of germ layer expansion truly is.
To visually reinforce what we have just learned and calculated, let us look at this cross-sectional diagram of a developing embryo. In the center, you see three distinct concentric rings representing the primary germ layers. The outermost blue ring is the ectoderm. Follow the arrows, and you see it gives rise to the brain and skin. The middle red ring is the mesoderm, which the arrows show developing into structural components like bone, muscle, and blood cells. Finally, the innermost yellow region is the endoderm, pointing directly to internal organs like the lungs and liver. This diagram beautifully summarizes the biological roadmap we have been discussing, showing exactly where each tissue originates. We have covered a lot of ground today, so let us summarize the most important ideas from this topic. First, we learned that the fate of germ layers is the ultimate biological roadmap, transforming a simple sphere of cells into a complex organism. Second, we mapped out the specific roles. The ectoderm forms the outer coverings and nervous system, the mesoderm builds the structural and circulatory components, and the endoderm creates the internal digestive and respiratory linings. Finally, we saw how exponential growth drives this process, and how even slight delays in cell division can cause severe developmental issues. You now have a solid understanding of both the biology and the mathematics behind embryonic development. Excellent work.